Hello and welcome everyone to this video podcast for Biology 211 Genetics Lab. In this video we're going to talk about Lab 1.3. The overall outline for this video is as follows. You have your lab quiz, which you've probably already taken, but if you're breaking it up between the three different labs, the next 5 questions can be done. If you remember, during that lab we learned about how we isolated DNA from plant tissues, and then how we took that DNA and we amplified it using PCR. And what we're going to do during this lab is learn about how we analyze the DNA product from the PCR. And one of the most common things to do is to run it on a gel and perform gel electrophoresis. And then we'll analyze these results in the gel. With this being an online lab, we don't have to wait for a gel to run. We can just look at what the results would look like or should look like. And then we're going to do the second round of PCR to increase the specificity and the yield. What we do from this lab, and it'll be again due on Thursday at 11:59, will be the worksheets 1.3 parts 1, 2, and 3. And then there's this gel electrophoresis simulation that you also must do. And you'll find that on Lab Exchange. There's also another thing on Lab Exchange that I put there. Initially I was going to assign it, but I know you have a lot to do, and so it's not an assignment, and so you don't have to do it. But I think it probably would take you 5-10 minutes, and I think it's useful to do. But it's not required. Again, just so you have a roadmap here of where we've been and where we're going with first module. We're right here, almost smack in the middle. So again, Lab 1.2, we're already done with that. And now we're taking that PCR results and running it on a gel, and then we're going to do the next round of PCR. What I didn't mention in the previous slides is that after this round of gel electrophoresis, we will analyze that in a couple weeks. Well, it says a couple weeks here, but really it'll be next week for the summer lab. Well, as I said before when we talked about PCR, we understand that you won't be able to physically do these methods and procedures during the online lab, but it's important for you to understand some of the basic skills and concepts. And gel electrophoresis is a very standard molecular biology technique that it's important for you to know, because one day you may be working in a lab and it'll be important for you to understand these features. So anytime we want to look at DNA, it could be from a PCR, it could be from a variety of other molecular biology techniques, but the gel electrophoresis works the same. We have a gel here, and then we put it in a gel box with buffer on it, and then we put the DNA in these wells. We hook the gel up to a power source, with the negative charge being at the end where the wells are, and the positive charge being at the bottom. By convention, the negative charged cathode usually has the black cord with it, and the positive anode is usually a red cord. What's important though is that the negative goes at the top and the positive goes towards the bottom. What color the cords are doesn't matter, except that by convention, that's the way it usually is. So we'll continue to follow that convention. Alright, so as we begin this discussion here, let's answer a few questions. What charge does DNA have? If you'd like, go ahead and hit pause and answer it, or you may have just answered it in your head immediately. Now remember, DNA that we've talked about in class has a negative charge on it. And has a negative charge on it because of all of these phosphate groups along the DNA backbone. Not just one, but they're all along the backbone here. And that gives DNA a highly negative charge. So what direction will it move in the gel? Feel free to hit pause and then come back. But DNA will move towards the positive end. Let's hope anyhow, because we put the DNA in these wells here, and if it ran towards the negative end, it's going to go this way. So that's why it's incredibly important that you hook up these cables correctly. If you hooked up the positive charge here, it's going to run the wrong way. If you hook up the negative charge here, it's going to run the wrong way. So it has to be negative, positive, so that, let me erase this or scribble this out so we don't get confused, so that DNA runs in this direction in the gel. As we can see down here, this is what the gel looks like after the DNA has ran through the gel. And the question here says, where would you find the largest DNA bands? Well, the figure kind of gives this one away. And yes, you would find the largest DNA bands near the top. Think of the gel as a sieve that allows smaller molecules to move through here. I had an instructor explain to me many years ago this concept by saying that pretend you had this big jar full of wiffle balls. And wiffle balls are just plastic balls that's often used to play baseball like in your backyard or something. And he said if you took a piece of sand that sand would go right through there because there's nothing impeding the movement of that sand. But if you took a golf ball, now a golf ball will eventually move through that tub of wiffle balls, but it's not going to move quite as fast. And that's what's happening here. The smaller DNA molecules represent the sand in my analogy, and so they move through the gel pretty quickly. And so they're going to be found near the bottom of the gel because they are not impeded as much as they move through the gel. However, larger molecules here are going to be found near the top because they cannot move through the gel as quickly. So what is the gel made of? Well, if you ever want to see a gel and we're back on campus, feel free to stop by and I will pour a gel for you so you can see what it looks like. In fact, if you want to, we could always just run a gel so you can see what that is all about. But when you hold it or you look at it, it looks and feels a lot like Jell-O. Don't eat it because it doesn't taste like Jell-O. I'm not going to tell you how I know it doesn't taste like Jell-O, but I'm telling you it does not taste like Jell-O. Agarose is a galactose-based polysaccharide. These agarose gels have a relatively large pore size, and this means that it's pretty good at separating DNA that's greater than 100 base pairs. Now, how well the gel separates fragments of DNA really depends upon the percentage of the gel. So in a 1% gel, which is a very standard percentage for gels, it is fairly reliable at separating out DNA fragments that are between 400 and 6,000 base pairs. And so the question is that optimal for this lab? To answer that question, go back to your manual and determine what the fragment size is that we expect. If the fragment size we expect is between 400 and 6,000 base pairs, then yeah, this is the right percentage of gel. And the answer is that this is the right percentage to use because the typical size of the gene that we're amplifying is between 500 base pairs and 2,500 base pairs, depending upon the species it comes from. The plant ones that are most closely related to kale, these are 1,000 base pairs, around 1,000 base pairs. So this is really what we expect. Now, that doesn't mean that's what we're going to see. Just using the evidence we have at hand, we would expect kale to be 1,000 base pairs. Just to go back to this slide here, the next slide we're going to talk about is how do we see these bands? They just don't appear as you're watching. It'd be kind of cool if as you were running it, you could see the DNA move. So how are we going to visualize the DNA in the gel? We usually use a dye that is called UView. It provides density to the sample, and that's really important. We'll talk about that in a moment, but that density allows the DNA to sink to the bottom of the wells and not float out. It contains this dye called bromophenol blue. It migrates with 500 base pairs of DNA, and you can see that with your naked eye. And so you know how far the DNA is moving in the gel, even if you can't see the DNA yet. I should also say that there is a second dye in it that migrates at 5,000. So when this is moving, in a gel, you have a general idea of where the 500 base pair and where the 5,000 base pair DNA should be. It also contains a dye that binds specifically to DNA. Now you can't see this until you put it underneath a UV transilluminator. That's why these first two dyes are really important, so you know the progress of the DNA in the sample. And so when you put it under a UV transilluminator, this is what you see, something like this. We used to use a dye that was mutagenic, and so we always wore gloves. So this one's not mutagenic, but even still, we always wear gloves when we work with any chemical that binds DNA, just as a precaution. When one runs a gel, they mix water and this dry powder, which is the agarose, and they microwave it in a glass flask here. Once it boils and it's in solution, it's poured carefully into this casting tray, which has a comb in it, and the comb is what's going to form the wells. Depending on how thick the gel is, it usually takes 20 minutes or so to solidify. You remove the comb, revealing the wells that you're going to use. You then take the gel out of the casting tray and place it into your electrophoresis apparatus. And then you put buffer on top of it. And then you add the DNA to the wells here. And remember, the dye that we were using allows the DNA to move to the bottom of the sample. We hook up our electrodes here to the correct ends, positive at the bottom, negative at the top. And then we turn on the voltage meter. Now, when you're running a gel, put the tip of the pipette that has the DNA sample in it right above it. It doesn't have to be inside the well. It just has to be above it. Because of the dye that we use, when it enters into this buffer, it just sinks straight to the bottom of the well. The reason I recommend not putting the tip of the pipette into the well is because you want to make sure that you don't puncture the bottom of the gel. Everybody who's ever ran a gel has done this. When you're pushing the DNA out of the pipette tip, make sure you do it carefully and slowly, because you don't want to have a big bubble in there come through there, because what happens then is that the DNA can flow out of the well into another well, and you'll see it too. It'll look like this big kind of poof of ink that kind of spreads between the different wells. One other thing I want to mention that I haven't mentioned before is what we add in this first well here. And in this first well here, we add what we call a molecular weight marker. Sometimes, in fact, often just in the lab, we call it a ruler or a DNA ladder. I typically call it a DNA ladder. This is really important. If you don't have this, you won't be able to effectively analyze your results. Because what this molecular ladder is DNA fragments of known size. And we know what these sizes are. And the one we use in this lab, you can look in your lab manual and it'll tell you the sizes. So for instance, if you look at this molecular weight ladder here, if you look at the band that is 3 from the bottom, that has the number 300 by it, we know that that is 300 base pairs. So we can make a rough estimate of the size of the band in our samples. So you see these 7 samples here, all about the same size. And so we would estimate that each of these samples is a little over 300 base pairs. We might estimate it at about 320 base pairs. This kind of gel, however, does not tell us the exact number of base pairs. We would need to use a different kind of test for that. We would never be able to say that this band is 338 base pairs, for instance. We would just have to estimate it at around 320 base pairs. So just a few basic rules of gel electrophoresis. Always turn off the box before you manipulate it in any way, before you add the buffer, before you add the gel, before you add the DNA. And then when you're done, turn it off before you take off the lid. You never want to have an accidental shock. Be sure the gel has solidified before adding the DNA samples. And this is a common mistake that happens early on. You can really tell when it's done because initially it looks like water, very transparent, and then when it solidifies, it's very opaque. This is an important step too. If you don't remember the order that you put your samples in, it makes it hard. Having a really well-organized lab notebook is very important. The simulation that you will run today actually has you do that before you can actually proceed. It has you put in the notebook, the electronic notebook, the order of the samples that you load. And it's important you do that correctly, because if you do it incorrectly, it will show you the wrong results, much in the same way as if you did not load it correctly in a real lab. Always wear gloves, just as a precaution. Today you don't need to wear gloves because you're not touching anything except your computer keyboard, which probably has more dangers on it than most lab benches, but that's another story. Put the leads in correctly. Everybody's done this once or twice. I promise you the leads are put on the wrong way. When I say leads, I mean the cables, mixing up the positive and negative cables. As I said, by convention, the black cable is the negative charge and the red cable is the positive charge. But really, I don't care much for thinking about the colors of the cable. I really want you to know that regardless of the color of the cable, the negative has to be at the top of the gel and the positive has to be at the bottom of the gel. The next 2 slides talk about the specifics associated with running the gel in our lab with the samples. I'm going to skip them in the video podcast, but they're in the PowerPoints for you to look at. So since you can't run a gel, what I want you to do is go to Lab Exchange. So go ahead and sign into that account, go to the Biology 211 Genetics Lab, and run this simulation. I think it's pretty good as far as simulations go. Just like you had with the pipetting simulation, it can be very specific. You have to make sure you open the lids before you take dye out. You have to close the lids. If you try to run the centrifuge without shutting the lid, it won't let you it won't let you run the centrifuge. So it has some of these basic safety precautions that a real lab would have. A few quick tips I want to pass on to you as you run this particular simulation, because I ran into some problems with it myself. Not really problems, I just didn't know where things were at. Make sure that as you're going through the simulation, that you go through each step specifically. Don't try to skip a step. Go through them just as the lab notebook on the right side shows in order. Otherwise, you might end up having to repeat the whole thing. One of the steps has you put the tubes in a centrifuge. And the step reads put in a centrifuge, and it's correct. You need to put them in a centrifuge. However, I couldn't find the centrifuge. It was nowhere in my visible view of the simulated lab bench. But if you close the notebook, there's a little thing near the top of the notebook for you to close it. And once it's closed, the centrifuge is under there. So don't spend time looking for the centrifuge. Just close that notebook and you'll see it. When you're done with the centrifuge, what you have to do is you have to take the samples out. And it says, place the samples. You have to take the microfuge tubes out and put them in a little rack. And so you open up the centrifuge and you see the tubes in the centrifuge and pick them up to put them in the rack, and they won't go in the rack. And it's very frustrating because try to specifically put it in the correct hole in the rack and it just doesn't work. But if you take the sample, and you put it in the blue box that provided the instructions to put the tubes in the rack, they'll fall right into the rack. So when they designed this, I guess they were off just a little bit of where that should be. So don't let that frustrate you. Just take the tubes out and place them not in the rack itself, but in the little blue box above it that provides the instruction. There are several steps with this simulation, just like you had with the pipette simulation. Go through all those steps, and when you do, I will get a message that you completed all the steps, and you'll get the points associated with this simulation. As I said before, there's this additional one. I wanted you to assign it, but I also wanted to be respectful of the time that you're already spending doing the worksheets and everything else. So you don't have to do it, but I think it's a useful assignment to do so that you can be confident that you understand this technique. The next slide is more specific to if you're in the lab actually doing these experiments, so I'm going to skip that, but leave it in the PowerPoint so you can look at it on your own if you'd like. Okay, so how are we going to interpret the results of the PCR by looking at the gel? Well, this is what our expected results should be. You would expect the fragment that you amplified through PCR to be about 1,000 base pairs. And so here we have our kale, we have our Arabidopsis, and then we have our DNA sample that's on the plasmid. And over here we have our negative control that we talked about. So remember, this is a positive control, legs 3 and 4 are, and then the water is the negative. And so the idea is this is what we would expect the size of the gene to look like, and we get about that size here with kale or cabbage. So when you're looking at this, let's answer this question here. What lane tells us whether or not we had contamination? We know that's the water. Because remember, there's no DNA template in this PCR reaction, so we would expect no DNA, and we would expect no amplification. If there was some sort of contamination, then we might see a band here. What combination of results would suggest a problem with the DNA extraction step? So remember, the DNA extraction was when we grounded up the plant material, the plant tissues, and isolated the DNA from it, and that DNA is what we use for the PCR. Go ahead and hit pause and think about that for a moment, and then hit play again to see what the answer is. So if we didn't see a band here in the kale or cabbage, but we still saw bands here in our 2 positive controls, then we would assume that something happened in our DNA extraction, because we used all the same reagents for the PCR in samples 2, 3, and 4. So if no result, no band here, that would tell us that we probably made a mistake in extracting the DNA. Now, what's likely to see here is, I'm just going to draw the band next to it. It's likely that the band we see for our kale cabbage is going to be less dense or less thick as for our Arabidopsis sample. We would expect that to be a much thicker and brighter band. And we would also expect the plasmid, and I'll draw it next to it here, also to be much thicker. Those were pure samples of DNA, and PCR just works better from that as opposed from extracted DNA. A band in which lane alone is enough to tell us that the PCR worked. Go ahead and hit pause and think about that for a moment, and then once you have an answer, hit play again. One would expect that the plasmid one is the one that should definitely work as long as the PCR worked. So we should definitely get a band there as long as everything worked well for PCR. You'd expect the one from Arabidopsis to work as well too, because remember that was the genomic DNA. But that has its own complications as well. So it's possible you would see a band here. But if you saw one here, you would say, yeah, PCR is a rock star and it worked perfectly well. If you're doing an experiment like this and you don't see a band here in lane 2, should your team give up hope? Well, of course not. And that's for a few reasons. Never give up hope, because the process of science isn't that it always works right the first time. It's that you investigate and figure out why something didn't work. Now, in this particular lab, we're amplifying DNA from raw tissue. Well, not raw tissue, but from tissue that's been grounded up. So while we expect to see a band here, it's possible it's not going to be very dense. It's possible that we won't see it, but it's still there. And so what we've done in the past in this lab is, even if a team had no sample here, we pretend like there is still sample there, and they proceed to the next step with the nested PCR. And after that, they find out that they really did have product there. And I'll explain that in just a moment when we talk about nested PCR. But never give up hope. But it's okay to get frustrated. So I just mentioned the nested PCR. So let's talk about the nested PCR in the context of this experiment that we're doing. In the second lab for this module, we did our initial round of PCR. And we used primers, and we called them degenerate primers. And they have less specificity for our target sequence. So they may bind here, but they might bind other places of the genome. But you do reduce the amount of DNA, right? Because remember, initially you have all of the cell's DNA as a target for the primers to bind to. And so these degenerate primers aren't as specific, so they should amplify your target sequence, but they might also amplify other regions as well. The nested PCR uses primers that are in between the first two primers, and that's why we call them nested, because they're in between. Now what this will do is amplify more specifically the targeted sequence. So let's go back and look at these degenerate primers. As I drew them here, they did turn out to be next to our targeted sequence. However, it is possible, as I said, that they could bind to other parts of the DNA, of the chromosomal DNA. And so this, I'm just going to draw them here. This is our first PCR at a target sequence that is not our desired one, somewhere else in the genome. Now when we do the second round of PCR, as I said before, it's going to specifically target these regions inside of the initial two sequences. The chances that your nested PCR will also contain the exact same sequences, like so, inside the initial mistargeted sequences is extremely rare. If it's not impossible, it's very nearly impossible for you to have sequences that bind here that are linked to our target sequence, but also have sequences inside that are the same. So this is one of the reasons we do the nested PCR, because it will increasingly specify the region we're interested in. And so that's one of the reasons, if we think about what I drew over here and here, that you might not see a very dense band in the gel, because you have these other areas also being amplified. So here you can see the primers we used, and this may help understand the mathematical impossibility that I was just talking about. The first primers we used here in our initial PCR is shown in blue, and the nested primers are shown here in yellow. Now the reason we don't just start with the nested primers, because you might think, why don't we just start with that? Well, if we only start with that, you're still going to end up getting some of that background, because while rare, you might find another sequence in the genome that matches these two. And so you might still amplify the wrong areas. But it would be incredibly rare in a DNA sequence to have this many specific nucleotides in a row that are the same. Same thing down here. So that's why the nested PCR will increase our overall yield at the end of the next step, which we'll show in the labs for next week. This slide here goes over pretty much what I just said on the past slide, so I'm not going to go over it, but it's in your PowerPoint slide, so you can look at it on your own. You can also look at it right here as well. So let me step back to this slide here. What we're going to do here, again, regardless if you found the band or not, we still take that sample of DNA after the initial PCR and redo the PCR with those nested primers. And so in the lab, you would have set up this next PCR. Unfortunately, there's not a good simulation like we have for the pipettes or the gel electrophoresis. So just go over these steps in your lab manual. And then in the next lab, for lab 1.3, we'll go over the results for the nested PCR. So looking to next week, which will have 3 separate labs again, but they're still all within the same module. You'll have week 2 quiz. It only has 20 questions because the next lab time is a time that will be devoted towards working on the outline. And so you'll work on your introduction outline in the next lab. And so that's a day devoted more towards writing. You'll want to make sure during that lab, you'll make figure 1 and write the components of the figure legends. For each of these writing assignments, you'll see there are very intensive grading rubrics. They seem kind of intimidating when you first look at them, but if you follow these rubrics, you should do well on these assignments. We'll also next week look at the results of the nested PCR and begin thinking about the sequencing and what the sequencing of that looks like and how to analyze that sequence. That's all I have for this video podcast for Lab 1.3. If you have any questions, make sure you ask me or your TA. If not, we'll see you in lab or lecture. Bye.